Tēnā koutou, ko Anne Schlesemanaho, and I'm at Manaki Whenua in Dunedin. And my work within More Birds in the Bush is focusing on forest bird population outcomes. And today I'm going to present two aspects of that work. So first of all, I'm going to briefly recap the framework that shaped a lot of the thinking around how food influences vital rates, given pest mammals in the system as well. And second, I'm going to present results from a field study that has grown out of that framework. And I would like to acknowledge the Parongia Te Aroaro Kahu Restoration Society, Doc Kirikiriroa staff, and Hapu around Mount Parongia for their support, as well as the tireless field workers, people in the lab sorting through inverts, and also a lot of the people behind the scenes doing the logistics to make everything run smoothly. None of this would have come together without that amazing team effort, so thank you. So in brief, what is the problem? And it can be summarized as the thermal squeeze. And Graham, who just talked, was the first one to show this based on data from Nelson Lakes National Park, using bird count data from the same locations collected in the 70s to early 80s, and then again in the 2000s. And I've just picked here two examples, bellbird and rifleman, and they were able to show that not only did overall abundance decline between those time periods, but particularly so at low elevation. And unfortunately, this phenomenon isn't just limited to Nelson Lakes National Park or to those two species. And Susan, Adrian, and John used the bird atlas data collected by the Ornithological Society right across New Zealand to show that bird occupancy of native forest birds decrease, particularly in more warm, floristically diverse forests. And this problem is, of course, of huge concern, but also may pinpoint towards where we could make the greatest difference for population restoration. As John touched on yesterday, the primary limiting factor of native forest birds are introduced mammals. I've put up here possums, rats, and stoats, but of course, these are not the only ones that cause havoc in our forests. As we again heard yesterday quite extensively, the resources in time and space drive the population abundance of these introduced mammals. And they, of course, prey on native bird populations, but also consume food resources that would otherwise be available for native birds. So they're predators and food diverters. When under high abundance of introduced mammals, food resources are likely to have only a minimal effect on vital rates of native forest birds. So by vital rates, I mean fecundity, juvenile survival, and adult survival. And that is because most breeding attempts are just terminated by predation. And so it doesn't really matter how many attempts are made or how big a clutch is, if the end result is that none of those eggs ever hatch. And similarly, juvenile and adult survival, depending on the species, can be significantly reduced by high abundance of introduced mammals. However, if those introduced mammals are suppressed, food resources may play a much bigger role in driving fecundity, juvenile, or adult survival. Currently in New Zealand, this is actually a bit of a knowledge gap that we don't understand that well in which way food resources influence fecundity or survival. And I wanted to address this knowledge gap using a large-scale field study and focusing particularly on the smaller rat-sensitive species. So these are mostly insectivorous passerines that are sort of smaller than about 35 grams. I wanted to use elevation as a natural productivity gradient and first answer the question of if actually along that elevational gradient, this presents a gradient also in food resources available to native forest birds. So does invertebrate biomass actually decline with increasing elevation? And I measured both ground-dwelling and flying invertebrates? And second, does that then also result in a decline of fecundity of native forest birds along increasing elevation? So rates of population change are generally strongly linked to fecundity, particularly of those smaller bodied species. So if you're working with a really long-lived species like albatross, survival kicks in and driving population dynamics quite a lot more. But with smaller species that have a shorter lifespan, fecundity can have a huge influence on population dynamics. And so I focus much more on fecundity. I chose 3 species as case studies, Miromiro tomtit, Titiponamu rifleman, and Korimako bellbird. All of these species have significantly reduced abundance and distribution since human colonization, but are also still actually able to coexist in forests with introduced mammals around. And therefore, they also have the potential to hugely benefit if we can get the pest control right and be able to restore populations. The other aspect that I took into account that those 3 species have slightly different life history traits and vulnerabilities. So for example, their nesting habits from being hole nesters or open cup nesters differ. And for example, their size is different. And so I wanted to know if actually the responses are the same across those 3 species or if they differ between the species. Unfortunately, despite huge field effort in trying to find bellbird nests, we were only ever able to track 1 female to a nest, and therefore I'm not going to talk much more about Korimako for the remainder of the talk. This is the place where that we used as a field study. That's Mount Pirongia near Hamilton in the Waikato. It provides an amazing elevation gradient, but as you can see, the lowland forest is cleared. So we tried to get the maximum elevation gradient that we could, but weren't able to go right down to near on sea level just because there's no forest left, unfortunately. We set up the study in 3 elevational bands. So at low between 200 and 300 meters, then a mid elevation 400 to 500 meters, and then a high 650 to 750 meters. Across these elevational bands, we sampled invertebrates in pitfall and flight intercept traps. These were run throughout 3 seasons, so spring, summer, and autumn, and always left open for about a month. And also we monitored rodents using tracking tunnels, and then stoats and possums using camera transects. Those camera transects we weren't able to stratify into elevational bands because then they wouldn't be independent anymore. So they were just randomly allocated throughout the site. Our initial field season was cut short, or actually rather started a bit later rather than cut short due to a delay in getting our wildlife permits. And initially we had set up 1 site that received predator control in the form of a bait station network that is serviced by the Pirongia Restoration Society, and a non-treatment site, which was the remainder of the Pirongia Forest Park. The second season, the remainder of Pirongia Forest Park received a 1080 treatment. So in the second season, the entire study site was under effective pest control. Due to the small sample size from the first season, I'm only actually going to present results from the second season and present all results together because our data show that pest mammals were effectively reduced across the entire study site and all elevational bands. First, let me take you into the field. So a combined 4,000 hours of field time made us experts at navigating supplejack patches and looking up, and up. So right at the bottom of your screen, you can see a person doing a nest observation, and many of the nests that we found would be high up in tawa trees, so more than 10 meters, sometimes over 15 meters, and you could just make out a changeover in incubation duties between riflemen, or if it was tomtit, the male calling off the female for feed. But we got just shy of 100 nests in that second season. And it definitely was those exciting days when you do actually find that nest or get to have your lunch in the sun with a beautiful view, and also get to finally sort through all of those invertebrates that were collected during all those times and see them all nicely sorted into the different size classes and orders. Back to my questions. So first of all, does invertebrate biomass decline with increasing elevation? What we expected was that we would see a seasonal trend, so a higher invertebrate biomass in summer compared to spring or autumn, and a decline overall between elevational bands, so higher abundance at low elevation and lower invertebrate biomass at high elevation. What we found though was that for flying invertebrates, we only found a seasonal pattern in biomass, so there was no elevational trend at all. And for ground dwelling invertebrates, there was an elevational trend, but only so in springtime. So you can see in green, that is the spring samples, and biomass actually decreases as you go up in elevation. So we had the strongest support for a model with an interactive effect for season elevation, only for the ground dwelling invertebrates, but only a seasonal effect for the flying invertebrates. So how did it look like for nest survival, and was it the same for both species? So we expected, given that there was a strong suppression of the key pests to low levels across elevation, that actually nest survival would be high, and actually would be high across all elevation bands, and that there would be species-specific differences between Titiponamu and Miro potentially, given that they've got slightly different nesting habits. And what we found was that the trend was the same actually for both species, which is quite cool, but also that nest survival was actually higher at high elevation for each species compared to the lower elevation. So we had the strongest support for a model with additive effects of species in elevation. As you can see though, effect sizes are small and confidence intervals overlapping. So it's the suggestion of a trend really. And how did this play out in the number of fledglings produced per successful nest? So we expected that there would be fewer fledglings at high elevation due to less food resources available to birds. And again, given the different clutch sizes, that there may be differences between the two species, Titiponamu and Miro. And what we found was that, again, we had the strong support for a model with an additive effect of species and elevation. So for both species, the trend was the same. There was no interaction between species and elevation, and that there was slightly fewer fledglings produced at high elevations compared to the other elevation bands. But again, the effect size was fairly small, and 95% confidence intervals overlapping. So to bring it all together, under successful suppression of rodents, possums, and stoats, we showed that for both species, there were the same responses, and we had support for elevation having an effect on it, with a suggestion of an increasing trend of nest survival with higher elevation, and fewer fledgling produced at high elevation, and fewer ground-dwelling invertebrates available in spring, which is when primarily most of the nesting activity happens. There was no trend at all for the flying or folivore invertebrates, and we think that that rain might be much more of a factor than simply just temperature, and rain is much more evenly would more evenly affect all elevation bands. The other part is that because our sampling window was a month at a time, that flying invertebrates may be either by themselves or through wind more easy to actually get up and down the mountain, and so any differences might be diminished through that longer sampling window. And again, to briefly sum up, so it's the breeding attempts that matter, which are primarily more food driven, as well as the outcomes. And so we have the first suggestion that actually there could be an elevational trend in nesting, or in fecundity, nest survival, and fledglings produced. But the study of vital rates of forest birds, particularly in those tall, complex forests, is really challenging, and yeah, requires quite a fit field team and loads of time. However, on the whole, it really, those vital rates allow to actually link habitat quality and like what drives those population dynamics, and you can actually understand much more of what is good habitat or versus bad habitat. And as an exciting next step, there's a master student that will use an experimental approach in supplementary feeding using a eco-sanctuary site and a non-fence site to tease out even more how predation and food interacts. Are there any questions?
Thank you so much, Anne. That was a great talk. You've had one question already, which was, did you measure temperature differences at your sampling sites?
Yes, yeah, we used little eye buttons at the same places where our rat tracking and the invertebrate sampling took place. And again, we can see that nice pattern of that high elevations are always colder than the lower elevation sites, but there was only about 4 degrees difference between our sampling sites, and actually a much greater seasonal difference between kind of from spring all the way through to summer and then back to autumn.
I've got a question. How did you know whether the invertebrates you were sampling, how did you know they were important to riflemen and tomtits in their diet?
Actually, I should say, James, the technician, and Scott, as well as Stephen Thorpe, were amazing at actually sorting through all of the invertebrate samples and sorting them into different size classes, so that we could actually look at what would be roughly what we would consider actually bird food versus bigger items that would still be eaten by possums or rats, and then also identified them to order. And through those orders, so we knew which orders were sampled and could go back to diet studies from back in the 80s and look at actually also later by other people, and where they documented what Miro Miro and Titiponamu fed on, and that actually matched really well, although of course it's mostly a general measure of available invertebrates that we did, but they were relevant to the bird species.
Cool. You've got a question from Colin O'Donnell. Do you think your altitudinal range was extreme enough to look at food availability?
That's a very good question. And most likely, I think if we would have been able to find a greater elevational gradient, I think the effect sizes would probably also be greater. We wanted to particularly work in those more floristically diverse forests, and we quickly ran into the problems of where all 3 species are present, but also as unfortunately John touched on, a lot of the lowland forest is gone. So finding that kind of having continuous forest cover in the North Island and then getting a long elevational gradient is challenging, and probably I think if we were able to carry this out for another 10 years, it would be really, really interesting to also look at that interannual variation.
I think we've got time for one last question. You've got one from Dave Kelly. Very interesting reduction in fecundity at higher altitude fits theoretical predictions, but the effect size looks so small, it is presumably of little practical impact.
Yeah, and I think that is a very good question. And one of the challenges that, or initially we were wanting to look much more at clutch size and also follow individual breeding attempts by having marked birds, but very quickly ended up with that logistical challenge of having not often that bigger windows to misnet birds and mark birds, but also resighting birds. So one thing that we weren't able to actually get at is the reproductive success per individual. So again, it'd be really, really interesting to do more follow-up work. And I think in terms of practical implication, the field is wide open. I think it'll be really interesting to dig more into how much food actually drives those vital rates.
Okay, thanks, Anne.